Hello folks, welcome to Cannabinoids and Proteins. It is my goal to survey the literature here and help you understand the mechanisms underlying cannabinoid treatment and all the different physiological effects that we've heard about but don't understand. Endocannabinoids are used throughout the body as a type of lipid signaling system. They are ligands for the cannabinoid receptors. Anandamide and 2-arachidinylglycerol are our main endocannabinoids of interest. Anandamide is named after the Sanskrit word ananda, which means bliss, and it is the main endocannabinoid active in the central nervous system, a high affinity cannabinoid 1 receptor agonist with high affinity at the vanilloid receptor, TRPV1. 2-AG is the most widespread endocannabinoid in the body, with moderate affinity at CB1R and CB2R and no action at the vanilloid receptors. G-protein coupled receptors are a device for cell signaling used for many different effects throughout cells, throughout the body. Specificity for this receptor type arises with the proteins expressed by the cell that associate with the GPCR. As you can see here, many different substances and things can activate the G-protein coupled receptor. Light, calcium, odorants, pheromones, small molecules, proteins. These are all used as ligands for the GPCR throughout many different cells and receptor systems. The G proteins expressed by the cell associate with the G protein coupled receptor and lead to the specificity of those systems. Cannabinoid receptors. Activation by an endocannabinoid causes dissociation of the G subunits, and the G proteins then affect other proteins. Kinases and transcription factors specifically of interest. Adenylyl cyclase is inhibited in the case of cannabinoid receptor activation, which leads to cyclic AMP-dependent processes being affected. Cannabinoid receptors versus transient receptor potential voltage channels. Both use G-protein coupled receptors to vastly different effects. When cannabinoid receptors are activated, G proteins dissociate and go on downstream to inhibit adenylyl cyclase, inhibit voltage-gated calcium channels, stimulate potassium channels, and stimulate mitogen-activated protein kinases. The TRP channel, a vanilloids effect, have a pore in them that allow influx of sodium and calcium. This leads to membrane depolarization and calcium-dependent processes. Cannabinoid and vanilloid receptors are co-expressed in many tissues, and anandamide is active at both, an endocannabinoid and an endovanilloid. This dual action distinguishes anandamide from other modulators. Here is a map of the presynaptic and postsynaptic neuron, and I've demonstrated activity of cannabinoid and vanilloid receptors. So what you get is neurotransmission. Here we show GABA and glutamate coming across the synaptic cleft to the postsynapse, which stimulates release of anandamide and 2-AG, which can then cycle back and inhibit further neurotransmission. This is what we describe as a retrograde neurotransmission. It sets up a loop that allows the neuron to regulate itself through the action of endocannabinoids. You see here anandamide is also capable of activating the vanilloid receptor, TRPV1, which will lead to depolarization of the neuron and will also halt neurotransmission. Extracellular signal-regulated kinase is an important enzyme in the context of cannabinoid receptor activation, and we can see effects on insulin control, neuroprotection, and synaptic plasticity, all through ERK-mediated processes that are stimulated by activation of cannabinoid receptors. Alzheimer's can be affected positively by locking up arachidonic acid in the form of 2-AG. This means that enzyme blockade and inactivation of monoacyl glycerol lipase can keep arachidonic acid locked up as 2-AG, which limits the possible production of eicosanoids. Those are known to increase beta amyloid plaques and tau hyperphosphorylation. So by inhibiting the enzyme, we lock up most of the arachidonic acid available to the cell as 2-AG and decrease beta amyloid plaque formation. Additionally, anandamide and CBD are shown to promote neurogenesis in response to beta amyloid insult, which suggests that these mechanisms might promote neurogenesis in a patient and help with underlying issues in Alzheimer's disease. It's also worth noting that CBD inhibits cytochrome P450. There's an interesting paper here that shows it's a mechanism-based inhibitor of CYP1A1. Structural analysis indicates that the bisphenol portion of CBD is responsible here. The intermediate is stable and locks up the cytochrome enzyme. This is a serious concern for patients on antidepressant or anticonvulsant medication and must be taken into account when patients are using CBD, tinctures, and other preparations for physiological effects. Interestingly, CBD and CBG both demonstrate anticonvulsant effects. So it's possible we can get patients off of pharmaceuticals and get them onto CBD and CBG to avoid potential problems with metabolism of their anticonvulsants. Thank you very much for your attention and look forward to the next one.